Good morning, students. Today we will be seeing the principles of biotechnology. What are the contents we have to study in this chapter is, first of all, definition of biotechnology, then the principles of biotechnology, tools of recombinant DNA technology, process of recombinant DNA technology. Now let us see the definition of biotechnology. Here, technology means a technique, bio means something living. So the definition of biotechnology goes on like this. It deals with technique of using living organisms or live organisms or enzymes from organisms to produce products and processes useful to human being. Or we can say biotechnology is the use of living system and organisms to develop or make up useful products. In a broad sense, we can say any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. So here we can say it is an integration of natural science and organisms like cells, parts of cells thereof, and also their molecular analog and products and services. Clear? Next, this is what I have told you. It's an integration of natural science and organisms, parts thereof, and molecular analog for products and services. Now, the principles of biotechnology. There are two major principles in biotechnology. They are genetic engineering and maintenance of sterile environment. On the basis of these two technologies only, we are moving on. But before going into the deeper sense of the technology, actually speaking, biotechnology was a process which we used unknowingly in our own homes, in our own houses, in our own kitchens. But we don't know that it is the process of biotechnology that is happening. So, what are those processes that were taking place in our kitchen? It is nothing but making of bread, curd. All this happens with the help of microorganisms only. Then, in a broader sense, we can say the fermentation process, the making of wine, alcoholic beverages, all these are fermentation. All the fermentation processes, the making wine and the beverages are all comes under fermentation process. Like that idli dough, what we make, we grind the flour and next day we are making it to ferment. Then only that idlis will be soft. Then cake making, like appams and all this, the basic principle is fermentation, and these were considered to be the traditional biotechnological process. From this basis only, we developed the modern biotechnology. So, in that modern biotechnology, we can divide that into two principles. It is actually working on two principles. First one is the genetic engineering, second, the maintenance of sterile environment. So, what is genetic engineering? We will see that. It is alternation of the structure of DNA and RNA into a host organisms to change the phenotype of the host. Then for that purpose only we are using genetic engineering. So it is a technique to alter the chemistry of the genetic material. What are the genetic material? DNA and RNA. And later, after altering the chemistry, we introduce these DNA and RNA into the host organism and thus change the phenotype of the host organism. So that's what shown in this figure. You are changing, this is a DNA molecule, a particular nitrogen base has been removed and it has been altered and it is introduced. See, you can see in the second diagram, the green color things are the original DNA, and into the original DNA, now you are introducing the DNA which you have cut from another organism. So thus you are altering the genetic makeup of the host. So this way, even by using a microorganism, we do this. Sorry. Maintenance of sterile environment. That is the second principle. The sterile ambience is very much essential because whatever we plan to grow only should grow in the medium of selection. So for that, 100% maintenance of a sterile ambience is necessary. That's why that particular microorganism alone will be grown. If it is done in a unsterile condition, what will happen? So many other microorganisms will also grow in the medium so that what you have decided will not take place. So it is to enable the growth of only the desired microbe or a eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccine, enzymes, etc. That is our aim, to make some product. So, to make a product, you have to produce our desired microorganism in large quantity without any what adultery there. Yeah. So it is necessary to maintain a sterile environment. Now let us move on to the basic steps that are involved in genetically modification. That is how we are going to modify the genetics of a organism. Number one, identification of the DNA with the desirable genes. Second step, introduction of the identified DNA into the host cell. Third, maintenance of the introduced DNA in the host and transfer of the DNA to its progeny. So these are the three steps which we use here. Okay. So first one, identification, isolation, and introduction of this into the target organism. And these are done by certain techniques. And for that, we use certain tools also. What are the tools we use? You know that no, whenever some engineering thing is done, let it be a electric thing or a mechanical engineer or automobile engineer, he needs certain tools to repair the automobile or to replace the parts of a automobile. It can be a scooter or a car, whatever it is. In the same way, here you are going to alter the genetic makeup. So to alter, you are isolating something, introducing that something into another organism and transferring that to your target organism. Clear? So to do these activities, you need certain tools. And the tools are restriction endonuclease enzyme, cloning vectors, competent host. These are the tools which we use. And the techniques for this are called recombinant DNA technology gene cloning, and gene transfer. Using these tools, we does three techniques: recombinant DNA technology, gene cloning, and gene transfer. So what is this tool? Number one, restriction endonuclease. It is responsible for restricting the growth of the bacteriophage in E. coli where it is extracted. The enzyme which adds, so on example, which adds deoxyadenosine methylase. This particular enzyme is added to the methyl group, and as a result, what will happen? The DNA can be cut by using another enzyme, restriction endonuclease. There are so many types of restriction endonucleases. R1, Echo R1, it is the Escherichia coli. So that particular bacteria is used as a restriction endonuclease here. RY strain, so many things. The detail, you need not study all the details, but just you know that this enzyme restriction endonuclease is used to cut the DNA at specific places. The restriction endonuclease must have palindromic sequences. Do you know what is a palindrome? The nitrogen bases will be arranged in such a way that this, the forward and backward way, when you read, it will code for the same nitrogen base. One simple thing is Malayalam is a letter. If you read from left also, it is MALAOALAM. If you read from right backwards, it's again MALAOAM. That is a palindromic word. Same way, here also the nitrogen base arrangement will be in the same order. Adenine, guanine, cytosine, thymine, and if you read it same way in the backward also, it will read adenine, guanine, cytosine, thymine. So that way of arrangement is called palindromic sequences. So the restriction endonuclease what you are using will be identifying these sites, and they will try to break the DNA or cut the DNA in these sites. So here example of the restriction endonucleases. See, the site is this. It is TT, AA, AATT. It is reverse AATT. So that is the site where it will be acting. So it RA restriction, RE has cut the DNA, and now the DNA is having sticky ends. Now the newly introduced nitrogen bases will go and occupy these areas. Okay, these unbroken edges are called sticky edges. If they are regular, it is called uniform edges. So it's clearly shown, it is cutting. So the fragment is now you are taking out the isolated portion into another organism's DNA where it is also having a sticky end. So this sticky end will attaches with that sticky end, and it forms a new DNA which is called the recombinant DNA. So this technique is known as recombinant DNA technology. Next is cloning vectors. We use certain organisms for the transfer of this. So vectors, normally we use the vectors like microorganisms. Okay, they are used as cloning vector. And these cloning vectors should have certain characters. It should be origin of replication. It should have a selectable marker. It should have a cloning site and vectors for cloning in plants and animals. These are the characters. So with this, I close today's class. And to talk about on the techniques, I will say few words about the techniques what we are using. The techniques, number one, recombinant DNA, then gene cloning and gene transfer we told. So for recombinant DNA, we need the tools restriction endonuclease to cut the DNA. Then we need a cloning vector to transfer the cut or the isolated DNA into another organism or the host cell. That is called the transfer. Before transfer, what we will do is we will multiply the recombinant DNA. So that is what is called as gene cloning. So on a recombinant DNA, they cannot multiply themselves. We have to inherit it into another organism and multiply it, or we have to link it with a medium so that it can multiply. That is called DNA cloning. And for after cloning, we transfer the huge mass into the host organism, can be more than one host organism, through a vector. So this vector is called cloning vectors. The commonly used vectors are plasmids. That is nothing but they are small circular, diagram is given here, small circular double-stranded self-replicating extra chromosomal material of a bacterium is called as a plasmid. So that is normally used as a cloning vector. Okay, and to talk about something about the cloning vector and all, I will stop sharing this and I will be sharing. Can you see the screen? Okay. So in this, the details of the techniques are given and their functioning is also mentioned very clearly. So this is what I have explained you. We require a vector. A vector is a plasmid, and these experiments were for the first time done by Stanley Cohen and Herbert Boyer in 1972. They done this experiment for encoding an antibiotic resistance in Salmonella typhimurium. It is used as a plasmid. The bacteria Salmonella typhimurium is used as a plasmid, whereby they try to create a antibiotic resistance in a host cell. So for this purpose, what they did is they found a gene which can induce antibiotic resistance. Then they tried to isolate it by what? Endonuclease. They started to cut out pieces of the DNA from a plasmid responsible for antibiotic resistance which involve molecular scissors. So that is the tool number 1, restriction enzyme. They are used and they are administered in the plasmid, which is having a DNA which has an antibiotic resistant property. So that particular DNA has been cut out in pieces using restriction endonuclease. Now, this desirable gene, you can call it as a alien gene. See, I have to incorporate with a special character, which I am not possessing. So when that character has been taken from a Y, that particular character will be a alien character to me, which is not belonging to my own. Clear? So that alien DNA is now linked into a plasmid which is acting as a vector. This plasmid will carry that integrated DNA with the antibiotic resistant property into the host organism. So linking of DNA, now linking means they have taken, isolated this DNA and they are now attaching the DNA into the plasmid DNA. So it has to get attached, it has to be pasted, and that is done by molecular scissor endonuclease cut that, and the gum which is joining the end is DNA ligase. So it will combine the DNA isolated with the plasmid, which acts on a cut DNA molecule and join their ends. New combination automatically joins, and what happens again? It starts replicating DNA created by us. I hope you can understand. Already there is a DNA in the plasmid. To this plasmid DNA, now they are introducing the isolated DNA bit, and this isolated DNA now incorporate with the existing DNA of the plasmid vector by DNA ligase. As a result, a new DNA is originated, which is called the recombinant DNA. Clear? So next slide, we will see the recombinant DNA now transferred to E. coli, because our aim is to create antibiotic resistant property in E. coli. So now the rDNA is transferred to E. coli and using DNA polymerase. This is again an enzyme. As a result, multiple copies of antibiotic resistant gene will happen in the body of E. coli. This is called gene cloning. So E. coli now will become antibiotic resistant. That was our aim. So thus it will become antibiotic resistant. So what are the basic steps involved? Identification of the DNA with the desirable gene, isolation, then introduction of the identified DNA into the host, maintenance of introduced DNA in the host, and transfer DNA into its progeny. So these are the diagrammatic representation of the steps what we have studied. What were the steps we have studied? That is the bacterium. It has a piece of DNA which you are going to isolate. And this piece has been incorporated with the already existing bacterial DNA. So now you get a recombinant DNA. So now this recombinant DNA multiplies. So gene cloning takes place, and later on it is multiplied and it is introduced into the host. This is an example of insulin, human insulin production. Clear? So same thing is represented in the form of a flow chart, isolating genomic DNA, then fragmenting the DNA, then you are screening whether it is correctly you have isolated. After screening, you are inserting that in a DNA vector. Then this vector introduces that into the host, culturing in the host cell, gene cloning, then transformation of host cell. So the first step, as we have studied, isolating genomic DNA from the donor. This is how we do it. Second step, fragmenting this isolated DNA by cutting using the molecular scissor, nothing but endonuclease. Then you are screening it, then inserting it. After inserting, you get the recombinant DNA that is introduced by microinjection processes that and all you will study later into the host cell. There it is cultured. Like many, it is introduced into so many media, so it is multiplied. After multiplication, it is injected into the host cell so that you can see the host cell having the desired character. So another thing is biotechnology products what we are getting out of biotechnology has got multiple uses. See, from microbiology, biochemistry, immunology, genetics, chemical engineering, cell biology, all these processes are involved in biotechnology. As a result of a combined effort, you get products like crops. You can introduce that technique into the crop production, in drug industry, in production of vaccines, and in diagnostics and livestock. So thus it has multiple area of implementation. So we told recombinant DNA technology we are doing with the help of the tools restriction endonuclease and restriction exonuclease. Then there is the polymerase enzyme, then ligases, vectors, and host. The enzyme that is used to cut the DNA is called the endonucleases, and that is the first step in the recombinant DNA technology. And in 1963, two enzymes were isolated from E. coli, which restricts the growth of bacteriophage. One enzyme is restriction endonucleases, and another enzyme added methyl group to the DNA. Thus, we have exonucleases and endonucleases. Totally nowadays, we have nearly 900 restriction enzymes isolated from 230 strains of bacteria. So the restriction enzyme actually belongs to nuclease class of enzyme. There are two types already we have seen. Restriction exonucleases, which removes the nucleotide from the end of the DNA. Restriction endonucleases, which cut at specific position of DNA. And each restriction enzyme can be named in a particular pattern. General term is restriction endonuclease. According to the role played by the restriction endonuclease on the particular DNA, you can name it. So that is a rule. The first letter of the restriction endonuclease should represent the genomic name. Second two letter represent the species name of the cell from which the enzyme was isolated. For example, if the restriction endonuclease is isolated from Escherichia coli, we represent the name as E echo, that is Escherichia coli, or I. It means that the enzyme is extracted from Escherichia coli of strand I. Clear? So this is how the restriction enzymes are named. And how it acts, the way it acts is, first it inspects the DNA sequences to find the specific recognition site. Then it goes and bind, and then only it cut the straight. Secondly, RE recognizes specific palindromic nucleotide sequences. Palindrome in DNA means the sequence of base pairs that reads the same on two strands. When orientation of reading is kept from 5 terminal end to 3 terminal or 3 to 5. See the example given here. In the 5 terminal, it is guanine, adenine, adenine, thymine, thymine, cytosine. When you read it from the 5 terminal, again, same thing. Guanine, adenine, adenine, thymine, thymine, cytosine. So this is the palindromic sequences. Then the restriction endonuclease cut the strand away from the center of palindrome sites, but between the same two bases on the opposite strand, leading to the formation of single-stranded portion of the ends. That is the sticky ends. So in the sticky end, hydrogen bond with their complementary counterpart. That is, if adenine is there, always it will pair with thymine. So whenever the adenine sticky end meets a thymine sticky end, it will get attached. And this attachment happens with the help of DNA ligase. So with this, I'm closing today's class. In next class, we can see further into the techniques.